Welcome to Dental Notes and Mnemonics. Today we are going to discuss about that why does normal blood not coagulate. So without any delay, let's get started with the video. Normal blood remains in fluid state due to balance between anticoagulant mechanism and the lack of activating stimuli. These factors can be classified into four main categories. The first is the intact endothelium. Second is the natural anticoagulants which is present in blood, fibrinolysis and continuous blood flow. They all work together to prevent the clotting unless it is needed.
Blood vessel is made up of three layers. The outer layer is known as tunica externa, which is composed of connective tissue, collagen, and elastic fibers. The middle layer is known as tunica media, and it consists mainly of smooth muscle cells and elastic fibers. And the internal lining of blood vessel is known as tunica intima. It contains endothelium, which is a single layer of squamous epithelial cells. Endothelium acts as a first line defense against the thrombosis. The endothelial glycocalyx is a gel-like protective layer lining the blood vessels, and it acts as a physical barrier repelling the platelets and clotting factors to prevent unwanted clot factors. Heparin sulfate in glycocalyx enhances antithrombin 3, which inhibits thrombin and factor 10, reducing the clotting risk. And by blocking the exposure of collagen and von Willebrand factor, it prevents platelet adhesion. It also senses the blood flow and stimulate nitric oxide release, which inhibit platelet activation and maintains smooth circulation. Certain anticoagulant substances are produced by endothelium and that prevent further clot formation. The key anticoagulants are nitric oxide, which prevent the platelet adhesion and aggregation, and prostacyclin, it inhibits platelet aggregation. Thrombomodulin binds to thrombin, converting it into procoagulant to an anticoagulant, and activates protein C, which degrades clotting factor 5A and 8A. Heparin sulfate enhances the action of antithrombin 3, which inhibits thrombin and factor 10A, preventing the clot formation. Tissue factor pathway inhibitor inhibits clot formation by blocking factor 10A and inhibiting the tissue factor and factor 7A complex. The endothelium work together to maintain a balance between the clotting and anticoagulation. Blood contains anticoagulant like antithrombin 3. It's a major anticoagulant protein produced by the liver. It inhibits thrombin and other clotting factor like factor 9, 10 and 11. Its action is greatly enhanced by heparin, which is why heparin is used as blood thinner. Other anticoagulant proteins which are secreted in the blood are protein C and the S system. These two proteins work together to inactivate clotting factor 5 and 8, which are crucial in coagulation cascade. Protein C is activated by thrombin when it binds to a special receptor called thrombomodulin, which is present on the endothelial cell, and protein S serves as a cofactor helping protein C function effectively. The balance between procoagulant and anticoagulant ensures that the blood remains in fluid, and they allow the formation of clot whenever it is required. Procoagulant are not always active. Most of them they circulate in an inactive form and are activated only when they are required. The clotting factors from 1 to 13 they exist as zymogens, which are the inactive precursors of clotting factors, and activate sequentially in coagulation cascade. Prothrombin factor, which is also known as a factor 2, is converted to thrombin only during clotting. Platelet remain inactive until exposed to the von Willebrand factor or collagen at the injury site. Tissue factor, which is also a factor 3, is exposed only when endothelium is damaged, triggering the extrinsic pathway. And calcium, which is a factor 4, and a factor 13, which stabilizes a clot only when it is needed. Likewise, anticoagulant, some always active and some activated when they are needed. The coagulant regulate clotting to prevent excessive thrombosis, and those which are always active are antithrombin 3, which is continuously inhibit thrombin factor 10A and other clotting factors. Tissue factor pathway inhibitor is also present continuously, and it prevents excessive activation of extrinsic pathway. Alpha 2 macroglobulin, it constantly inhibits thrombin and plasmin. And the anticoagulant which are activated when they're required is protein C and protein S. They're activated by thrombin and thrombomodulin complex to degrade factor 5A and 8A. Plasminogen activators convert plasminogen to plasmin, which degrades fibrin, but only when the clot breakdown is required. Plasmin also remains inactive until it is needed for fibrolysis. Normal blood flow is laminar, meaning it moves smoothly in the parallel layers with minimum resistance. This is crucial for preventing the thrombosis, as laminar flow keeps the platelets and clotting factors evenly distributed, reducing the risk of clot formation. Also, it reduces mechanical stress on the blood vessels, preventing endothelial injury that could trigger clotting. So, we have gone through all the factors which work together to prevent unnecessary coagulation while allowing controlled clotting when it is needed. Thank you for watching this video. If you found this video helpful, please like, share, or subscribe for more videos like this. See you in the next video. Till then, bye bye. Take care. Have a nice day.